right. So let me introduce Mark here for you. Mark is a principal and general manager of Stadys Incorporated. Prior to joining Stadys, he spearheaded an award-winning quality improvement program for an international manufacturer, generating millions of dollars in profit. Mark offers a diverse array of experience in process development, quality assurance, marketing, purchasing, and general management. He's also the co-author of 2 books, DOE Simplified, Practical Tools for Effective Experimentation, and RSM Simplified, Optimizing Processes Using Response Surface Methods for Designing Experiments. He's also published numerous articles on DOE. He's a guest lecturer at the University of Minnesota in the Chemical Engineering Material Sciences Department and at the Ohio State University Fisher College of Business. Mark is a professional engineer and a certified quality engineer, and he also holds an MBA from the University of Minnesota. He also has a bachelor's in chemical engineering from the University of Minnesota. So during the presentation, if you have a question for Mark, please use the GoToWebinar, enter a question for the staff section in the control panel, and we will be monitoring those questions that come up and answer as many of those as we can during the session. Any other issues you have, please post those in either the chat or the question area, and we'll try to address those as we can. So without any further delay, here is your presenter, Mark Anderson.
Thank you, Sam, and thank you, Stat Division, and thank you, American Society of Quality, for sponsoring this presentation. It's been my pleasure to be a part of ASQ since the 1970s, and I feel like when I got my certified quality engineering, it was like a big light bulb coming on for statistical tools, and in particular, my main focus is on design of experiments, or DOE. And for this talk, the main tool will be DOE for QbD. What's really kind of interesting and even ironic is that what's leveraging QbD at this point is Indian generic drug makers who have been incented, you might say, by the United States Food and Drug Administration to make use of better tools to assure the quality of their product, and these come under the banner of QbD, or Quality by Design. For example, the credit for the graphic here goes to a few from the country of India in Ahmedabad, an article called Total Quality Management, and it shows in this graphic how what we're going to do is come up with a design space within which the control can float around and never go outside of specifications. What's interesting is that these ideas need not be only for pharmaceuticals, but really can go beyond into any manufacturing industry. And therefore, I felt it was good to provide a briefing to my fellow quality professionals about these developments that have occurred in the field of pharmaceuticals. Now, just recently, within the last 6 weeks or so, there was an article in Chemical and Engineering News that talked about design of experiments making a comeback. And the comeback is into the process industries, which are chemical, pharmaceutical, and so forth, but it's really the pharmaceutical industry and the advent of this technique of QbD that's generating a lot of interest in quality tools and in particular design of experiments. And so here we have a couple of comments from the article that drug firms and their suppliers say they have stepped up the use of design experiments in development work. And Robert Dugood at Albany Molecular Research said, it is definitely increasing, particularly in late-stage development. And he went on to say that we are proposing to all our clients that we do DOE analysis for robustness testing processes before they go into commercial production. So these are music to my ears as a quality professional that's been working for many years on trying to prevent problems rather than just try to inspect and correct. And then Dugood goes on to talk about how this QbD guidance has really affected what they're doing in this pharmaceutical area. So that's news hot off the press from Chemical Engineering News, and it did catch attention from a lot of people. So it's moving out from this field of pharmaceutical into the chemical and hopefully beyond that into other manufacturing industries. So let me begin then by laying out the agenda. What I'll do is start by talking a bit about what really is the quality by design concept and what is design space in particular and how can that help us, what's in it for us. And from there I'll go on to talk about a couple of different levels of QbD, one that involves the use of confidence intervals or CIs, mainly for functional operations, and then a more severe approach that imposes tolerance intervals or TIs on finished product to make sure that we provide an adequate setback within which we can be safe in terms of floating a bit with our manufacturing. And then there's issues in terms of doing a proper designed experiment and sizing it appropriately, which basically is like a power calculation for sample sizing. But we have to use some specialized tools to come up with an adequate sizing. So let me begin then by talking about the quality by design concept. So working within the design space is not considered as a change. So this is the magical words here where a pharmaceutical manufacturer such as these Indian drug makers can show to the FDA that they've got a region that they can stay within that assures the quality of their product and they can be approved for selling in the United States and in Europe. What I like about this definition is it talks about multi-factor testing and the fact that variables can interact with each other to create unexpected outcomes. And this is where the design of experiments will come into play. So what I'm saying is that this merits attention by all quality professionals, not just in the pharma. And that's why I felt it was worth providing this briefing. Now just a little sidetrack on the terminology of quality by design. It turns out that there's a book by Duran that's titled Quality by Design. That's a landmark in the field of quality. However, in this book, as Ron Sneed noted in his Pharma Evolution blog, where did the term design space come from anyway? Duran never actually talks about design space per se. And Ron Sneed goes on to credit the ideas of design space more to the developers of design of experiments and responsiveness methods, which are Ronald Fisher in the fields of agriculture in the 20s and 30s. And then in the chemical industry in particular, George Box developed RSM. And then, beyond that, other stat-savvy scientists in the field of pharmaceutical that recognized the power of DOE and RSM and began to develop these concepts that became the design space for quality by design. So it's a little bit of background here then on what is QbD, the angle from FDA, the idea of design space, and so forth and so on. Now I had to go back and refer to Duran, who is a graduate of University of Minnesota and had a long and distinguished field in career in the field of quality. And what would he view is going on here with this QbD? Well, really, it's the pressures from the US FDA and the European regulatory agencies that are driving this in pharma. That's what's pushing them and levering them to make use of these quality tools. If you're not in a regulated industry, how can you get people interested in taking a more rigorous and more disciplined approach to quality? Well, that might be difficult, and Duran had kind of a cynical point of view of how that might happen. He said, pray for things to get worse. So, sorry to throw a little note of cynicism in on this, but as a matter of fact, sometimes the quality professionals are the ones that have to light a fire under the manufacturing people to get them to start working on preventive activities such as this QbD. All right, now moving on to some nuts and bolts here. Let's talk about functional design space for unit operations, where we're going to frame a sweet spot with confidence intervals. Now what I'll do along the way here, especially as I start getting into more of these nuts and bolts, is I'll stop and ask for questions on some occasions But if I get on a roll, I've alerted Sam that he shouldn't hold back from interrupting me and saying, hey, hold on a second, we've got a few questions. So I can certainly stop for a bit here and take some questions as we go. But let's continue on at this point and get into some nuts and bolts. So let's talk about design space, but in particular what I call functional design space to differentiate it from the final product definition. Now, as a chemical engineer, and I worked in process development for many years and actually worked on manufacturing process improvement as well, what I learned early on is that you couldn't improve the entire manufacturing site all at once. You had to focus yourself on a given unit operation, and typically either it would be the first unit operation in a series, or it might be a critical unit operation, for example, one that was a bottleneck. And we actually would focus on a given unit operation and really dissect it and come up with maybe an early version of the so-called design space. The idea then is to focus on one unit operation and try to control it well enough that we can develop a fairly tight span on each of the input factors, let's say plus or minus 2 standard deviations, and control it tight enough that even if it floated around a bit, it would still stay within a region that would provide quality product. So this control space is very tight. However, due to drifts in raw materials, due to aging of equipment, due to natural shifts from people that change the process a little bit, this control space may float a little bit, but as long as it stays inside this yellow region, which is a design space, then we're going to make quality product. So the trick is to be able to map out this design space or sweet spot and then make sure that it's we're far enough inside of it that we won't have much of a chance of ever going out of spec on this particular process. So that's the basic idea that we're going to apply here. I have a couple of examples of where we've applied responsiveness methods to map out a region and find the sweet spot. And then what you'll notice is that inside this yellow region are gray areas where we pushed away from the outer boundaries by the half width of the confidence interval on the mean for the response. And this example is from aerospace, and I talked about it in 2011 at the ASQ conference in Pittsburgh. And so if you're interested, you can go back to that briefing from 2011 to see how I actually did this. But again, what we have here is a region, which I'm drawing out here with my red marker, that is the sweet spot. Okay, so this area here is sometimes called an overlay plot, where we've shaded out any results, and there's two different responses here that don't meet specifications. So all of the parts outside of this red box that I'm drawing lines out from, this area on the outside of this region does not meet requirements for the unit operation. Okay, so here then in the middle is what I would refer to as a sweet spot. As long as we stay inside the sweet spot, the process will meet requirements and we can continue on to the next unit operation. However, because of the fact that we did an experiment, which really was just a sample of all possible outcomes from the manufacturing process, we actually do not know for certain where that outer boundary is. So therefore, to be conservative, we add a confidence interval that pushes in. I'm going to erase all my markings at this point, and we're going to push in from the edge of the sweet spot then by the confidence interval. So here we have the lower requirement on the first response, and here we have the upper requirement, and here we have the upper requirement on the second response, and the upper requirement and the lower requirement. And then these shaded regions represent the confidence intervals. We're pushing up a little bit from the bottom, we're pushing down a little bit from the top, we're pushing over to the right from the left-hand boundary, and to the left from the right-hand boundary. And that leaves us then a smaller region that is more conservative. And so I would refer to this as a CI framed design space. And again, it's CI, or confidence interval, that we're framing at this point, because we're really just interested in the average performance of this unit operation being within the guidelines. So this is actually a big step forward from simply working within the sweet spot by adding the confidence intervals to frame it. So let me stop at this point, Sam, and see, do you have any questions that have floated in at this point?
I haven't seen any questions come across yet, so feel free, if anybody does have a question, to use the ask. Wait a second, one just came through right now. Hold on a second here, so I can make that bigger. So someone asked a question, if you use a tolerance interval instead of a confidence interval, then does the whole design space, will it disappear?
Yes, that's a good point. It turns out that to size an experiment for a tolerance interval is going to require many more runs. And so that is exactly the reason why for the unit operations leading up to the final production, it's better to make use of the CI, the confidence interval, because it's, of course, easier to pin down something on average than it is for individuals. And we'll see later in some further slides how many more runs are actually going to be required to do the more rigorous TI rather than the CI.
Okay. And then another question that came through, it says, instead of design space for each unit operation separately, would you recommend an overall design space combining multiple unit operations to explore between-step interactions? I think that, yeah.
I think so. I mean, it seems to me that what we're going to be doing is something that's going to be suboptimal because it's practical to focus, step by step on each unit operation, either starting from the top and working your way down, or starting from the most critical bottleneck operation and working your way back upstream and then downstream. At some point, you begin to group unit operations, and finally, you might even draw a box around the entire building that's producing a given product. And this is the type of thing that I would work on with my team in manufacturing process improvement. There wasn't really one right way of doing things. And so sometimes I would think of it as I'm a little guy running on the outside of a ring, and inside is a giant sumo wrestler. And at some point or another, I'm going to have to dive into the ring and grab a hold of this guy and try to wrestle him down. But it doesn't really matter, where I grab him. It's going to be a very tough battle. At some point, I just have to jump in and try something. So that's kind of how it went with the manufacturing process improvement projects that I would work on with the quality professionals and process engineers. But I think to start with, it's good to focus on a very critical unit operation, and definitely you can make some progress there and then move on from that point.
All right. Well, I think that's the extent of the questions that have come through so far.
Okay. All right. I will continue on then. And I just again want to mention that there is a briefing that was provided at the 2011 International ASQ Conference. And of course, this was also under the umbrella of the Institute for Continual Quality Improvement, or ICQI, which has been a group to bring back some of these hardcore tools that seem to slide away for a number of years from the annual conference. I have another example that stems from a study that was presented in the International Journal of Advanced Manufacturing Technology, and it has to do with something completely different than the prior one, which was aerospace. This one has to do with a milling operation, and there were two key responses that they were trying to work with to reduce the tool wear, of course, and also to maximize the material rate that they were coming in with. And so response surface method was run, and then they did a numerical optimization using desirability as an overall utility function, and they were able to come up with one combination that's shown here that was the ideal, most desirable point. But of course, we have to realize that we're not going to be able to operate this machining or milling operation right at this point all the time, and so it would be good if we gave ourselves some space. So they developed some specifications on material rate and on tool wear, and these created a window, which is the sweet spot. And then inside of this window are superimposed confidence intervals to provide a more conservative region that we hope to stay within, and then have very little chance of the process ever going out of control. And so these are just the two examples then that show you how we can frame the space and then fill in the frame with a confidence interval, one out of aerospace and the other from manufacturing, electrodischarge milling. So hopefully this gives you a picture of where we're going here. By the way, this fancy picture underneath here is a picture of desirability, and you can see how the desirability pushes up within the sweet spot region, which is projected on the bottom here. So that's just kind of a fancy picture to make it look more compelling, but really the picture that's helpful is just a simple overlay plot that shades out the parts that don't meet spec and then push in with the confidence interval to give us a conservative operating region. So what we've been doing then is we've been recognizing that statistics means never having to say you're certain about anything, and whenever we collect up data and say the average of the data is such and such, we also want to recognize that there's an interval around that within which if we were to go back and collect up more samples, we might fall, or basically that interval will float around up and down based on the different samples that we take from the process. So to try to convey uncertainty, then we come up with some type of a distribution like this normal curve, and here we have the limit of specification listed, and if we run right at that limit, then of course 50% would be out of specification. Now just a little sidetrack anecdote. When I worked for a chemical company at their manufacturing facility south of Chicago, I went to the plant after achieving my certified quality engineering. I was working with them to try to improve their quality, and they had a process I call goalpost quality, where they had the upper spec limit and the lower spec limit, and anything that fell between those two limits was a field goal. And so what would happen is after a while they would drift towards one end or the other. Let's say that they drifted towards the upper spec limit. So now let's say that the actual process is operating right at the spec limit, and this I saw happening quite often. Well, you would think that 50% would be out. However, whenever they got a high value, they would retest it, and because of the variation in the test itself, they would actually test the product back into specification. When I say test the product back into specification, that's a joke, because of course you can't test something back into specification. But they thought they had done so they would pass the product. Now, if the product, the second test, also came out of spec, they would then again test it, and they might test it many times until finally, by chance, one of the tests would produce a product that came below the specification limit. So actually, there were many processes when I went down in the 70s to this plant south of Chicago that were operating just like this, where the mean was right at the specification limit, and they were truly producing over half outside of spec, which is kind of scary. So that isn't what we really want to do, is it? It's not something that we would want to do by design. So this is why we're going to put in this confidence interval and push down from the spec limit far enough so that only a small percentage is likely to be above the spec. And so the confidence interval, the half-width of that interval, provides us with conservative framing or padding to reduce the chances of product on average actually falling outside of spec. So I think that's pretty obvious, and this is the first part of my talk that I actually presented in 2011 and briefed on that. So that actually is a review of prior presentation. Now the new part that I came in with to our talk in 2013, and I have to mention at this point, and I was remiss in not saying at the beginning, that the co-author of the presentation is my business partner here at Stadys, Patrick J. Whitcomb. And actually, Pat is the fellow who does all the heavy number crunching. And I just try to present it and make it more simple and easier to understand. But Pat is the heavy lifter on terms of the number crunching. So, so this is a newer part that Pat developed, and he was able to stand on the shoulders of some other excellent statistical experts who developed techniques to come up with tolerance intervals for design of experiments matrices. And so that's what we'll talk about next. So the idea then is to push up another notch from confidence intervals to tolerance intervals, and at the very end of the process, when we're producing the final product, we want to be more conservative and even say that all the individuals that we produce will fall within specification, not that we're just in specification on average. So let's talk about this more conservative way of framing the sweet spot. And then you could work with either 90, 95, or 99%. Well, we're going to work with 99% of the population, so we're going to go with 95%.
Welcome to GoToWebinar, Web Events Made Easy.
What we're going to do is use response surface methods to model the responses as a function of the process factors, and then we will apply tolerance intervals to back away from the edge of this sweet spot or provide a buffer so that ultimately we'll have a design space that's very conservative and will meet the requirements of the FDA. And so we're going to have to consider how we size this experiment. As somebody asked earlier, wouldn't the sizing for the TI cause the whole area to shade out? And that's correct. If you have an undersized experiment, there will be no window because the TI will actually close off that window. So we have to be careful to do an experiment that's large enough that can support the imposition of a tolerance interval. So what I'll do is I'll go ahead and dive into this case study, and before too long I'll stop again for questions, but it's usually easier to talk about something specifically rather than in general. So I think you'll find this easier to launch off of when we have the questions come in. So this actually is a tabulating process, and it turns out that, as I mentioned at the beginning, most of the work that's being done in this field is coming out of pharmaceutical, and in particular from these Indian manufacturers of generic drugs. They're the ones that are under the gun to come in with these cheap pharmaceuticals, but pharmaceuticals that are providing the efficacy that are needed, not just in terms of the active pharmaceutical ingredient, the API, but also in how quickly they dissolve, which is called the rate of dissolution, and also the physical properties of the tablets have to be good. In other words, they can't fall apart. They have to have what they call proper friability, and also the shape of the pills have to be held up. So they have a number of parameters, and it seems that they're primarily focusing in on the tableting processes, and that may be the most difficult part of it, is coming up with pills that will not fall apart and that at the same time will dissolve, in the proper period of time to deliver the drug. So here's kind of a typical study that we've been working with these Indian pharmaceuticals that involved the response surface method design, exploring two we had to block off some parts of the experiment because they would just produce bad product. And to do that, we imposed.
Welcome to GoToWebinar, Web Events Made Easy.
To understand, if you think of a process to make breakfast cereal, for example, what's going to happen here if we look at two factors on making breakfast cereal, which is the temperature and the time, is that if we go too low on time and too low on temperature, you'll just get mush. And so this lower corner has to be eliminated from our experimental region. At the same time, if we go too high in time and too high in temperature, we'll burn the product. And so then again, we have to block off a region. And so these multiple linear constraints give us the opportunity to create a diagonal space like this that cuts off the high combinations and the low combinations, or any other part that doesn't work for us. Now, as a chemical engineer, of course, I was mostly concerned about this combination of high time and high temperature, because very often it would create an exothermic runaway reaction, which was something you didn't want to have happen. So we are going to impose these multiple linear constraints to frame our experiment to begin with, and then we'll systematically vary the inputs and measure the outputs, which are shown here. So the two factors that we're going to work with are granulation and lubrification. The responses are dissolution, friability, and hardness of the pill in terms of kilopons. So mathematically, we would express the inputs as x sub 1 and x sub 2, and the outputs are y sub 1, y sub 2, and y sub 3. And the goal of the RSM is to create a model that predicts the response y as a function of x. And so this y equals f of x is the product of the response surface method, which is a model. And we're going to develop models for 3 different responses. And then we'll actually map those out, shade out what doesn't meet requirements, and create the overlay plot. And then inside that, we'll frame it with the TI. Okay, I'm going to stop here for a moment because I've been talking for a while and just see if there's any questions about this next part that I've just talked about. Anything come in, Sam, on the question box?
I haven't seen any additional questions come across yet.
All right, no problem. I don't mind that. And Sam, if questions come in that you're not able to cover during the presentation itself, will you email those to me?
Yes, I'll make a copy of them and we can share them. One question came across and said, what about collinearities when working with funny shaped spaces?
Right, well what will happen is you'll get sometimes a very narrow track. It could be that we have a diagonal that's extremely thin here, and then it's going to be hard to differentiate the input factors from each other. But we've got a process that I'll show you that can size overall even when you have a high degree of collinearity. Normally if you try to use power calculations, it'll fall apart because eventually it'll become so collinear that you'll never be able to get above the alpha level on the power calculation. But because of the technique that I'm going to show you that uses overall sizing using fraction of design space, we can overcome that. And the models themselves, even though you can't differentiate the terms very well individually, the overall model can be very significant and very useful. And as you know, Bach said, and everybody quotes him on this, and may he rest in peace, passing away this spring. But all models are wrong, but some are useful, and that's a good mantra to go by. All right, well let's move on then, and let's take a look at the initial operating window for this tableting process. So as I mentioned, we applied these multiple linear constraints, or MLCs, to block off the high combinations over here in the upper right corner, and also the low combinations have been blocked off as well, so that we didn't even do any experiments in this region. The actual experiment would have hit these vertices here, and so where I put these X's are points where we would have actually done combinations of the granulation and lubrification and measured the responses. And typically you get centers of edges, typically the overall center point, and then many times you'll get what are called checkpoints that fall between the center point and the extreme vertices. And what we used for the experiment actually was an optimal design strategy. And just FYI is we would recommend something called an I-optimal design that would populate the region with points, something like what I just showed here. There has to be enough experimental points that are unique to be able to using optimal design. So that was done, and then using the models, we shaded out anything that fell outside of 0.5 friability, outside of 75 dissolution, and outside of 10 hardness. Okay, so then in this yellow region is an operating area or window or sweet spot, and in theory, if we could stay in there, we'd be fine. However, of course, this window that we've framed out is based on sample data, and we don't really know for sure that where these lines are drawn is where they're going to end up being over the long haul. So we have to apply some statistical sensibilities here and realize that if we run right at the edge, we're going to probably be in bad shape and really be producing a lot of off-grade. So what we're going to do then is we're going to move away from those boundaries by some interval. If it was a functional process unit operation, then I might say you can get by with the confidence interval. But let's assume this is the final manufacturing step. So now we're going to apply the more rigorous. And here again, what I'm going to go by is 95% confidence that 99% of the portion of population will stay within the data limits. And this seems like it's going to be good enough for the Food and Drug Administration and the European regulatory. Surprisingly enough, in my discussions with the FDA guy in Washington, D.C. that works on generic drugs, is I'm usually trying to convince him to be more rigorous in his standards, in that he feels that it's already pretty hard for some of these generic manufacturers to do what they're being asked to do, and to try to push them even harder is unrealistic. So that isn't exactly what I'm interested in hearing as a quality professional, but I have to realize that you can only go so far at any given time. So if we can achieve this level of quality where they're pushing in by the tolerance interval and doing it with 95% confidence that 99% are falling within those limits, I would say that that would be quite an accomplishment. So now we get into some issues for the actual computations. And as many of you may recall, there's a fairly simple approach to developing a tolerance interval. Thank goodness that others have worked on this and put together tables so that people like myself that are chemical engineer MBA but not PhD mathematician or statistician can easily construct these intervals because somebody else has put together a table. And the table that we want to work with is the K2 table. And with the K2 table, you can look up a factor then that represents the half-width of the tolerance interval. So, of course, the first thing we have to do is calculate out the standard deviation S, and then look up the appropriate K2. There's a book by Hahn and Meeker that have every imaginable statistical interval that you could ever need, and I would recommend the Hahn and Meeker book on statistical intervals if you're ever looking for information on how to construct these. So, so using this K2 table, then we can develop a plus or minus around the mean, with K2 being the factor times S, and so then this breaks down to Y bar plus or minus D, where D is a half-width of the tolerance interval. And so there's a half-width there. And so that tells us that we can be confident with 95% that, say, 99% of the population will fall within these limits. Now, it turns out that to work on a confidence interval, as we've recognized from the outset, is going to require more data. Another way of looking at it is that the confidence interval is going to be a lot narrower than the tolerance interval. And here's the equation for the confidence interval, y bar plus or minus t times s over the square root of n. As I mentioned, for the tolerance interval, we're going to use this K2 factor. Now, looking at the degrees of freedom which affect the value for t and K2, you can see that, of course, at the very low degrees of freedom, both the t and the K2 are quite large. As we increase the degrees of freedom or sample size, then they both converge down to the z value of 1.96 for the 95% interval. But in between, whereas where we're likely to be, let's say at 10 degrees of freedom, what you'll see is that the K2 definitely is much larger by an appreciable percent. So that means that we're going to have a bigger sample or have wider requirements to be able to accommodate the tolerance interval than we would for the confidence interval. And so that's something that we're going to have to keep in mind. It's going to be a lot harder to come up with a properly sized experiment. Now, here's where the rabbit gets pulled out of the hat by the statisticians and mathematicians. And this is detailed in this article on using the intervals for prediction, a tutorial on tolerance intervals for ordinary least squares regression, published in Chemometrics and Intelligent Laboratory Systems in 2007 by Gries, Langhans, and Vandergroup. So they did the pioneering work that allowed us to calculate the tolerance interval for a designed experiment such as a response surface method. And here is the calculation. What it boils down to is a quantity that ends up being multiplied by S. And this is known as the TI multiplier, or TIM. At least that's what we're calling it. Just a little side joke on this is TIM brings to mind TMI, which is too much information. So I won't belabor this, but there are equations that can be programmed in that are making use of the X matrix, which is the input factor levels in coded form, and inverse normal distributions and all kinds of good things like chi-squared distributions and so forth and so on. But it's actually something you can compute if you have the proper equipment. So now let's take a look at what happens with a simple tolerance interval that's just based on a single sample versus what you need to do with a designed experiment. And what you'll notice is that there's another little inflation that goes on here. In terms of a designed experiment, we have to apply a little bit more of a padding to our edges to accommodate a designed experiment and to push in properly from that point using a tolerance interval. So that adds even more requirement for us to have to do a bigger experiment. So the tolerance intervals that cover a multi-factor region are wider than those at a single point. And hopefully that would be intuitive that that would be the case, but exactly how much you can calculate. So I'll just stop again, Sam, and see if anybody has any comments or questions about this part.
Yes, one question that came through. You mentioned a book. I believe it was the book by Hahn and Meeker. What's the title of that book?
You know, I believe it's Statistical Intervals. I'm looking over at my shelf, but unfortunately it's in another office. I think it's over in Whitcomb's office.
I think that's right, too. It is called Statistical Intervals. So, and then another question. It was a few slides back, but someone said the experimental points that you just showed were numerous. Is it economically possible to do that many experiments, especially if you add replicates to the experiment?
Probably not in many cases, particularly for these Indian drug makers. So we have a new innovation that I'll just briefly touch on that I'm hoping to come back, if not next year, the year after, and talk about. But because of the pushback from the actual manufacturers that don't want to do so many runs, we came up with a technique that is sort of akin to ASTM E1169 ruggedness testing for analytical methods that presumes that you're already operating at a point where there's very little effect of any of the key factors. And given that, you can do kind of a go, no-go type experiment where you can apply factorial design rather than response surface methods. And the difference that you use for the sizing ends up being, approximately 1/4 of the specification width, which is actually a larger difference when you go in and do the calculation that facilitates a smaller experiment. So we've come up with a protocol, but it's predicated on the idea that you can assume that we're already operating at a point where it's fairly robust and that we won't see very large effects. And that allows for then a smaller experiment that can still accommodate the tolerance intervals. Being a chemical engineer, a certified quality engineer, being an MBA, of course I would much rather have a more rigorous experiment such as a response surface method like I'm showing here. And also as a person who takes pharmaceuticals, I'm taking a statin drug and so forth, I would really like the manufacturers to go all out to make sure that the product is meeting requirements. But again, as a practical matter, trying to get these manufacturers to even do a designed experiment is kind of hard. So I think we've come up with sort of a workaround that's maybe somewhat of a compromise, but still much better than what they had been doing before. So that I can only say is that there's some further developments coming along on that, so stay tuned and watch for my next presentation. But what I'm showing here really is certainly the rigorous approach. So what I've done is I've taken the numbers, and actually Pat was kind enough to do this for us, and converted it into a graph that makes it more visual, what the rigorousness is in terms of moving from doing a confidence interval, which is the bottom line here, using the t-value times 1 over the square root of n, and then looking at the single sample K2 line, which is the olive colored line here, and then even more difficult is the tolerance interval that goes with a design experiment, which is the TIM, the tolerance interval multiplier, and you can see that in the scarlet. And then, of course, they drop off as the number of degrees of freedom go up. But at 10 degrees of freedom, you can see that there's a big increase from trying to do just a confidence interval up to a tolerance interval on a single sample, and then there's a further increase that's required if you're working with a multi-factor experiment. Okay, so now let's just look at an example here in the last 10 minutes or so, and I'll give you an example of a TIFrame design space. So what we're going to do is try to develop an experiment that will be large enough to accommodate pushing back from the edges of our sweet spot and still leave a design space left over that it doesn't get completely shaded off. And actually the number of runs that are going to be required are going to be dependent on the ratio of the signal, the half-width d, and the noise, which is the standard deviation. And you may be familiar with this idea of signal to noise to calculate power for a sample size, for example. Now, we have 3 different responses, but we can focus on the one that is going to be the hardest, which has the lowest signal to noise ratio. So that will dictate the size of the experiment, the dissolution. So now here is the actual experiment. It was done via I-optimal or IV-optimal. We needed 6 model points to fit the overall intercept, the 2 main effects, the 2-factor interaction, and the 2 squared terms. So we needed 6 points at a minimum. We also added 5 additional checkpoints for lack of fit, and then out of the original 11 points, we replicated 5 of those. So where you see the red dots are where the combinations were run. And you can see that the center point was run 4 times, and then a couple other points were replicated here in the corners. This is the experiment in randomized run order. And then what we're going to do is we're going to size this experiment, and we're going to use a technique called fraction of design space to do it because of the collinearities. We can't just apply the power calculation directly. Now, I've talked about FDS sizing in my prior 2011 talk, so you can go back and refer to the 2011 talk, and also this talk here, which is available off the Chemical and Process Industries Division, or CP&ID site, Whitcomb's talk on sizing mixture and RSM designs. We'll explain more about this FDS. So to keep it simple, let me just say that with the fraction of design space, what we want is to get at least 80% of the space to meet the requirements that we put in for precision. We want to be able to estimate within a certain half width, and we want to be able to do so within most of the design space. And to accomplish that, you have to have a proper number of runs or combinations of factors. So it turns out that when you're sizing using FDS, you're going to use similar guidelines as you would with power calculation. Typically with power, you say we want to have at least 80% power. You normally don't want to try to get to 100% because that would be an infinite number of runs. So when we do the power calculation, we do the sizing calculation on this using the normal functional design sizing for CI, it turns out that we have to be more rigorous in our requirement on D. The D of 9 is so easy to accomplish using CI sizing that we reduce it down to 3, and then we're able to get an FDS of 96%, which is still plenty fine. So this is actually a more demanding specification, like an internal spec, that we put in just to try to crank down the FDS somewhat from 100%. So what is FDS? Just a quick sidebar here. It's the fraction of design space that falls within the requirements that we have for the precision. So here's a really simple case where we have one factor X, and we're measuring one response R1 or Y1. And we do a fitting on this, and we find that it follows a straight line, which is the middle part here. Well, naturally, of course, if we draw in the plus or minus interval on this, it forms this kind of flared out line, which is a dotted line. Now, let's say that we really want to stay within this plus or minus D that's shown here. Then if you look at this very closely, you'll see that the red line, which is the desired precision, is accommodated only over the middle part of this design. Let's say about 50% of the region is being estimated to within the precision that we're looking for. Once we get outside of the zone here and go to the higher or lower end of the factor, the plus or minus goes outside of the red line, and that's not desired. So the FDS is actually this part here where we're able to estimate to the precision we desire. In other words, the red lines are outside of the dotted flared lines. And so that's what we actually measure when we do the FDS. So here's a FDS on this curve here, and the FDS is 51%. So only 51% in this little case study I just did falls within the desirability for precision. Okay, so now let's go back to our example with the tableting, and let's say we want to use a TIFrame design space. So now we're going to size this by the TI multiplier, which you remember is a lot harder to hit than the CI. And it turns out that even going back to the difference of 9, which is actually a pretty big signal, we're unable to get any FDS. It's 0. In other words, the design that we started with here is not enough. We don't have enough runs to be able to do a TI-framed sweet spot. So what are we going to do? We're going to have to do more runs. How many more runs? Turns out that only 7 more points, which we create by over-specifying the I-optimal model points. So we go from 6 to 13 on the model points, 7 more than what we really need at a minimum, and that's enough to get the FDS high above 80%. So that we can be fairly certain that when we impose a tolerance interval, it won't block off our sweet spot. So here's the final design, which has 23 runs. It's got the 13 model points, which are 7 more than we started with, to accommodate the sizing for the TI. And then we go ahead and collect up the 3 different responses and analyze them and model them. And these are the contour graphs for the 3 different responses, fit to a second order quadratic equation. And then if you look carefully, there's actually contours in here that are red. And so we want more than 75%. So in the dissolution, the part that I'm shading out here outside of the red line doesn't meet requirements, but the part that's inside that shaded area does. On the friability, here's the red line here, and we want it to be less than 0.5%, which is the blue region here. So this part doesn't meet spec. And then the hardness has to be above 10. Here's the 10 line here, and this part here doesn't meet the requirement. So imagine we take this plot and put it on top of this plot, and put this plot on top of this plot. So we've got 3 of them, one on top of the other, which is how we used to do this back in the good old days using overhead view graphs. We'd actually shade out for each individual view graph what didn't meet spec, put it on top of the next one, and put it on top of the next one, and hopefully there would still be a window inside there. And there is. So here is a window here. The original window was out here someplace, right? So this was the original window, but then we pushed in by the tolerance intervals on the friability, the dissolution, and the hardness. And so now this is actually our design space here. So this is the QbD design space, the part that's pure yellow. All right, so we went through this whole process of selecting key factors, setting up a response surface method to generate the critical responses, and then resize the RSM to accommodate the tolerance interval, ran the experiment, did the fitting, put the tolerance interval in, and we ended up with the design space that's shown here. And this is set up with tolerance intervals for 95% confidence, which is alpha 5%, and that we get 99% of the individuals, falling within these ranges. How many more runs are going to be needed for the TI versus the CI? Well, it depends on how many factors you have. But here we can see that we added 7, which was a 44% increase on the first example. There was 4 factors, 10 extra runs, and so forth. So you can see that to accommodate this TI framing, we do have to do a larger experiment. And so in summary then, we need to size the experiment appropriately for the purpose. If it's functional design, we'll use confidence intervals. That isn't so bad. But if it's final product and using tolerance intervals, we're going to have to make a larger experiment to accommodate that. Because of the TI multipliers being larger than those for the confidence interval. The sample size increases, of course, then we get the degrees of freedom, and it becomes less of an issue. But for the typical sample sizes or degrees of freedom that you have for an experiment, it does become an issue. So the tolerance intervals will require a larger experiment than the confidence intervals. And so in conclusion, what we're going to do here is try to extrapolate from the pharmaceutical area this quality by design space concept and try to apply it to other manufacturing processes. And not only at the very final stage where we apply the tolerance interval, but in the buildup of the product, we can apply a little less rigorous approach using confidence intervals to make sure each unit operation will, on average, produce good product. And then finally, at the end, by the time we get that accomplished, hopefully things are pretty well lined out, and we can do the TI framed sweet spot. So here is a picture that comes from FDA, and basically it's showing the control space, which is a plus or minus 2 standard deviations on each of the inputs, for example. And it really would be a circle, probably, like this. And then this can float around in the space, or the whole frame actually moves around because of drifts in raw material and things like that. But either way, you've got this region here that's a buffer so that the chances of it ever going out of spec are much reduced. And then as far as what next, I gave you a preview of that, and that is if we can assume that there's no effects of any practical importance, then it's possible that we can make use of factorial design rather than response surface methods, which is simpler and requires fewer runs, which saves money. And then we're going to size it by some fraction of the overall spec range, let's say approximately half or 1/4. And this will be very much like this ruggedness test, E1169. I was a member of the committee that rewrote this into a standard some years ago. And so I'm very familiar with this approach. And I think it can work, but it does require a big assumption at the outset to facilitate the use of these fewer run factorial designs. So therefore, to be really conservative, I would advocate the use of response surface methods, and then finally the TI framing on the product. So I threw a lot of information out there, especially the second part of it where it got into the nuts and bolts. Maybe we have a few minutes for questions, Sam. I'd be certainly willing to hang on for it. But that concludes my talk.
Yeah, that'd be great. And thank you, Mark, so much for that really interesting, informative talk. It's very relevant, especially in the pharmaceutical industry. I know right now this whole area of quality by design. So we really appreciate you taking the time to present for us during our webinar. A few questions that came across. One was, and it related back to when you were setting up the number of runs you need to have in an experiment to do this tolerance interval approach. Was instead of increasing the sample size, would you recommend simulation tools such as Monte Carlo simulation to assess process robustness, assuming you have a good model?
The use of simulations are very helpful to reduce the number of hands-on builds or experiments, and providing that you can verify the simulations, through some type of confirmation run. It's becoming more and more commonplace to use such things as finite element analysis on the design of turbine engines and so forth, and then applying against the simulation an experiment such as a response surface method, perhaps an optimal design. Typically, you end up over-specifying the model points so that you get a better coverage of the space, or you use such things as Latin hypercube designs. And then you pull out what's called a transfer function from that, which simplifies things quite a bit. And then from that, you then develop a sweet spot. But at some point or another, you're going to have to actually make the product and be sure that it does fall within that window. Monte Carlo simulations can come into play there in terms of the simulator. I haven't actually seen it in this arena, because here you're doing hands-on work using a pilot plant or an actual manufacturing tableter or the like. But I think as we start to think about beyond pharma, perhaps this could be something that would be computer aided design and design of experiments.
All right. And how about one more question? Well, let's see. Look at the questions here. Well, I think just a couple questions to wrap up here. One is someone asked if you can get a copy of your slides, and another asked if the webinar is being recorded. And the webinar has been recorded, and this will be saved and published out on a YouTube channel in the near future. So it'll be available for future use. And then I would recommend, if you'd like a copy of the slides, just to contact Mark directly. And you could ask him for a copy of the slides. Mark, if you want to show your contact information again there. And there's his email address. And then you could reach out to Mark, and he could provide you with that. Okay. So I think that's it for the questions, and we're right here about 5 minutes after noon Eastern Standard Time, so I think it's time to wrap up. I appreciate everyone who registered for the webinar, and glad you could attend. I hope you found this useful. And we would like to encourage you, if you are a member of the Statistics Division, thank you for being a member. We really appreciate that. I serve on the leadership for the Statistics Division and the communications team. And if you're not a member of the Statistics Division, we really encourage you to join. So you can join the ASQ, and the Statistics Division dues are only additional $10 a year to be part of the Statistics Division. We also have a LinkedIn page on LinkedIn group if you're a member of LinkedIn, and we also have a Twitter account, so you can follow us in a lot of different ways. We're trying to use more modern social media to stay connected with our members and get the information about statistical thinking and quality out to everybody.